Time. All right, here we go. A couple more people popping in. I see Roger and Lily, but I can't type anymore. So, this is the one. Bioenergetics. Ladies and gentlemen, for some reason, one of the lowest scores, the two sections that are kind of the lowest on the Keystone, are homeostasis and transport and bioenergetics. And bioenergetics being one of those. And so you would think that bioenergetics is this huge topic with lots of information that's really complicated and really hard to understand. Not so much. So I'm not sure why people tend to do poorly on that the questions for bioenergetics, but we're going to change that this year, at least with those of you that are here. And it looks like there's somewhere between 15 and 20 of you here. So first, thank you for coming. It means a lot to me to know that you guys want to do well on the Keystone. I mean, it can't be just for the bonus points, right? So, desktop. You are now seeing my desktop. On my desktop is the bioenergetics eligible content, the stuff they are allowed to test you on. And if you look at it, there's only three things. There's describe the fundamental roles of plastids, for example, chloroplast, in the mitochondria and energy transformations. Compare the basic transformation of energy during photosynthesis and cellular respiration, and describe the role of ATP in biochemical reactions. And I'm going to say it, and I want you to yell it out so mom and dad wonder what's going on. I say energy, you say, right. You say ATP. And somebody was like totally making fun of me for that today, but the person will remain nameless, although I think they might be here right now. So a couple more people, Lily, Connor, Anna, and look, there's Mrs. Smith. The wonderful and delightful Mrs. Smith has joined us. So yeah, I mean, this is all we have to do, guys. This is not hard, right? Just these three little things. So we're going to demystify this tonight, and we're going to try and make it so, like, you don't miss these questions. And I think the one big rule of thumb to take away from this is anytime you see the word energy, look for ATP. Anytime you see the word ATP, look for energy. If you do those things, you're probably going to get the question right. So, sitting down here on my paper, let's go to the paper view. Here it is. This is the first graphic organizer we made. And I want to draw your attention. I'm going to mute the mic 1 second and cough. Sorry, I don't want to cough in your ear. If we look down here, we've already done this. We did this the first night. We have the chloroplast drawn. We have the things that go in the chloroplast. We come out of the chloroplast with sugar. That goes over to the mitochondria. And then that goes up here to ATP, where we can use ATP to do work. So we've already done it. We've already talked about it. So, like, come on. This is easy. All right. Graphic organizer. You should have a piece of paper with you. So be ready to write. I think the other night when we did chemical basis of life, it took us about 40 minutes. I don't think this one's going to take us 40 minutes. So I want to start up here in the corner and just get the basic rule of thumb down. We are starting with light energy. Is light energy the only place life on Earth can get energy? And the answer to that is simply no. It is not the only place that life on Earth can get energy. There are bacteria that live in the bottom of the ocean that get energy from chemicals and things like that. So there's chemosynthesis and other things happening. But the majority of what they're going to ask you about and talk to you about is this idea that life on Earth gets its energy from light. So we have light energy. What's the first thing that has to happen? We need to take that light energy and change that to stored chemical energy. All right, so we take that and we store it. So now we go from light energy to stored chemical energy, and then we need to go and be able to transform that into usable chemical energy. Right? And so right here in these parts, These are the different transformations that take place. And so I'm just going to block that off because everything that we're doing tonight, everything that we're talking about is that. How do we go from light energy to stored chemical energy to usable chemical energy? That's it. See a couple people in the chat room said they have to go get paper. Yeah, guys, if you want to turn in the graphic organizer you write down, you have to have a piece of paper to write it down. But that's how you get your points. So everything we do is this. Now, what is the equation? So I want to write up here across the top equations. And so I have a paper in front of me. I kind of wrote this out how I want to do it, and I'm changing it as I go here because I think I could lay it out a little bit better for you. So let's write the equations. What is the equation for stored chemical energy? So notice I'm going to kind of write this straight across from that. And here's the other thing. You don't have to memorize this equation. Your background knowledge, drawing on life experiences and the things that you know and have been told your whole life, you can pull this equation out of thin air. So, if I asked a third grader, what things do plants need to grow? They would say sunlight and water. Somewhere between third grade and ninth grade, they probably would throw in their CO2, because you all seem to have come to me understanding that plants need carbon dioxide, they breathe in carbon dioxide, and they breathe out oxygen. And so if you have that, they breathe in carbon dioxide, they breathe out oxygen, and they need sunlight and water, you have the equation for photosynthesis. All you need to do is go, okay, they need carbon dioxide, they need water, right? Somewhere along the line, they need energy coming into that system, and then they breathe out oxygen. And so all we're going to do is add to that C6H12O6, which is one thing you maybe would have to learn. That's a plus sign. Wow. Goodness, shooter. So we could balance this. We could put a 6CO2 and a 6H2O and a 6O2 if we wanted to. But I just want you to get the bare bones of this. I want you to make sure that you remember this. And again, I don't think you need to memorize it. I'm sorry, I can't speak. Long day. I don't think you need to memorize it. I think what you need to do is understand it. Hey, I know plants need carbon dioxide and water. I know they need sunlight. So here's my carbon dioxide and water. They need energy. They make C6H12O6, which is sugar. So down underneath of that, we could write sugar, or we could write glucose, whatever you want to call it. But you should know that molecule by now. We use it all the time. And then they give off oxygen as a waste product. So how do we go from light energy to stored chemical energy? Right here. That's how we do it. Now, how do we go from stored chemical energy to usable chemical energy? Now, think about this. What is stored chemical energy? It is C6H12O6. So what do you think I need to put with that to break that up? Well, it's just the exact opposite. So to understand photosynthesis, all you need to do is be able to do the opposite of that and go backwards. So C6H12O6 plus O2 yields CO2 plus H2O. And we could balance it, but you can do that next year, Mrs. Urban. And of course, energy comes out of that equation, right? Bam. Done. We should probably write down where these things happen. So where does this top equation happen? Waiting for an answer I'm never going to get, right? So this top equation happens in the... Here, can I do this real quick? This top equation happens in the chloroplast. And this bottom equation happens in the mitochondria. Which you should know. I'm going to draw a line blocking that off. There we go. Bam. Chloroplast, mitochondria. Now here's an interesting question for you. Both of these, the chloroplast and the mitochondria, are organelles. Mrs. Smith and I were talking about this today after school at the bus stop, waiting for you guys safely, as we were safely assisting you onto the bus. How, if these are both organelles, these are both membrane-bound organelles, how does a prokaryotic cell do either one of these? All right, how does a prokaryotic cell do either one of these? Let's talk about that at the end. All right, stick around. There's the cliffhanger for the evening. The cliffhanger. How does a prokaryotic cell without membrane-bound organelles do either one of these processes, store chemical energy or get to usable chemical energy if they do not have membrane-bound organelles? Bam, just like that. All right. Let's jump down and talk about the chloroplast. We have a chloroplast. We drew it in class. It's a kidney bean looking thing, although my kidney bean looking thing isn't very good. And inside that kidney bean looking thing, there are these stacks of something. And I'm just going to draw a couple here. All right, so what do we have? This little stack is called the granum. All right? Each one of these little discs in the stack is called the thylakoid. And this takes us back to how artists draw these things and how we can tell if they're drawing a chloroplast or a mitochondria, because you'll look for stacks in it. And then inside of the chloroplast, all this fluid is called the stroma. So one of the things that I didn't like when we showed you, when you guys filled out a graphic organizer about this, is they had one granum, and then they had this big circle over here where the Krebs cycle happened, or the Calvin cycle happened. And what I didn't like about that is I don't think it really kind of conveyed what was going on. So what's going on here is there's two things happening. And so I'm going to draw a 1 right here, 1, and I'm going to draw a 2 right here. So one thing is happening inside the thylakoids, and one thing is happening right outside of it in the stroma. Don't think it's like shipping stuff way out here and then shipping stuff way back in. It's just bouncing back and forth between the membrane of the thylakoid. Okay. We're getting somewhere. What's number 1? Number 1 is the light-dependent reaction. So over here, I am going to draw one disc of the thylakoid. Wish I would have made it a little bigger. So, okay. That disc is, what's happening in that disc is part of the thylakoid. What's happening is the light-dependent reactions. So they're going to be happening in that disc. And then next to that, right outside in the stroma, we have another thing happening. I'm going to draw a circle with some arrows to show you that this is kind of circular. And that is going to be the Calvin cycle. Okay? Good. So, in the light-dependent reactions, we have a couple things happening. One thing going into the light-dependent reactions is water. So water goes into the light-dependent reactions. And oxygen comes out. So I want you to think about this, because like I said, we want to demystify this. We want to make it look like, hey, this isn't hard. Why? If water goes in and oxygen comes out, what have we not accounted for? Well, we haven't accounted for that hydrogen. Like, what happened to it? We need to account for that. So we're going to do that right here. Coming from the Calvin cycle back into the light-dependent reactions is NADDP+. That's a D. You just have to believe me. So that is coming from the Calvin cycle into the light-dependent reactions, NAD+. There's another thing coming, and that is ADP plus P. And so that is also coming in here. Now, I want you to think about this, because it's really not hard, right? This NADP plus and this ADP, adenosine diphosphate plus P. Well, if we add that P to it, we have what? Adenosine triphosphate. So we have ATP. But we have to have energy to do that. Well, where does the energy to do that come from? Well, this is the light-dependent reaction. So not only is H2O coming in, but also light is coming in, right? So light is coming in too. So we have 4 things coming in. 1, 2, 3, 4. We have light coming in, water coming in, NADP plus and ADP plus P. We use the light, the energy from the light, to take the hydrogen off the H2O, and we add that. I could have drawn this better. I apologize, but we add that and make NADPH, which goes into our Calvin cycle. We also use that energy to make, you guessed it, ATP, which also goes into our Calvin cycle. That's it, right? Not that hard. We take the hydrogen from the oxygen, we use the energy from light to add that to these to make NADPH and ATP. We send those over the Calvin cycle. So we have a question. Here's a question I asked you earlier. So I'm going to do this like kind of weird arrow here. Big question. So why not just use this ATP? This was an essay question we did. Why not just use that ATP? Like, seriously, we just made it. Like, what are we fooling around for? Why not stop there? Just use that ATP. And the reason for that is because that ATP isn't where we want it to be. Like, we want to store that ATP so we can use it another time. We don't necessarily want to use it right here, right now. You know what I mean? Like, if Snicker bars only let you eat Snicker bars as they came off the line, what good would that be? You'd have to drive to the Snicker bar plant to get a Snicker bar. And you could only eat it right there and right then. So instead, what do? We package it up and we let you take it wherever you want and eat it whenever you want. Same thing. We need to take this ATP, package it up so we can use it whenever we want. So how do we do that? Well, think about what we have going on. We have H2O coming in. Go back to our equation up here. We have H2O and energy coming in already. What are we missing? CO2. So if I add above the Calvin cycle CO2 and have that come in, do you see it yet? We need to make a sugar. What's a sugar? C6H12O6. Well, we have carbon, oxygen, and now we have the hydrogen on this NADPH. So if we take that hydrogen off and add it to this, we can trap some sugar. So coming out of the Krebs, or the Calvin cycle, I keep saying that wrong. Hopefully Mrs. Smith is gone so she doesn't bust my chops for that. We have C6H12O6 coming out. All right, giving a pause here for people to catch up. Had nothing to do with me wanting to take a sip of my coffee. All right. So I probably could have drawn that over a little bit and made it a little easier to read, but anyway. So look at what we have. H2O plus CO2 plus light energy yields C6H12O6 plus oxygen. Right there, we have the entire process done. So what have we done so far? In the first 18 minutes here, we've gone from light energy and we've gone to stored chemical energy. Now what we need to do is figure out how to go from stored chemical energy to usable chemical energy. All right, a couple more notes on this piece of paper before we flip it and get to that. What is this process called that we just talked about? If we are in the chloroplast of a plant cell and we are in the thylakoid doing the light-dependent reactions where we use H2O and light and give off oxygen and make NADP and the ADP plus P and the NADPH and ATP, what is this whole thing called? This is photosynthesis. This is photosynthesis. Major important thing here, right? Exclamation points or whatever they are. Photosynthesis. So now what we need to do is go take stored chemical energy, and we need to make that into usable chemical energy. And this one I think people struggled with a little bit more than photosynthesis, but I hope by the end of this you're going to go, no man, this is easy. This is cake. All right, I tried to leave that up. A point of interest, if you're having trouble getting this all written down, When the video's over, the live video will go, the recording will go up, and you can just scrub back and get it written down. If you're watching this it's not live, you can always pause it and do it. So that's gone. Bam, just like that. And we're coming over to the... Oh, no, we're not coming. You want a blank one. I got to rewrite it for you, right? All right, here we go. Blank piece of paper. So, what's the question? On the top of this one, let's go stored chemical energy to usable chemical energy. That's what we're doing right now. Stored chemical energy to usable chemical energy. And we are starting with glucose. We're starting with C6H12O6. This is glucose, for those of you that forget. Glucose. We're starting with the sugar. How do we do it? Well, we got to get to the mitochondria, but before we can get to the mitochondria, there's a step that has to happen. And that step is called... I can't say it and write at the same time. That step is called glycolysis. We're going to put glycolysis in a little box here. First thing that has to happen, we got to get to glycolysis. Now, we're getting close to answering that question I asked you in the very beginning of this. I asked you if chloroplasts and mitochondria are both membrane-bound organelles. How then, right? How then does a prokaryotic cell do it? So we're getting close to that. In glycolysis, there's two kind of caveats or rules, right? We're going to go in one of two directions. The first direction we're going to go to is with oxygen present. So if we have oxygen present, we can go this way. If we don't have oxygen present, so I'm going to put no O2 present, we're going to go this way. How many ATPs does glycolysis make? It actually makes, we get something out of this. We get out of this 2 ATP. So we make a little bit of ATP, which is really interesting because we actually make 4 ATP, but we have to use 2 ATP to even do this process. So if I said to you, if you pay me $2, I'll give you a job that makes you $4. You didn't make $4 at the end of the day, you made $2. And that's kind of glycolysis. It costs us $2 to make 4, so we end up with a net gain of 2 ATP. So let's start over here. If there is no oxygen present, what happens? We end up with one or two choices. We get our 2 ATP, and then we make one of two things. One thing we might make is lactic acid. The other thing we might make is alcohol. Simple enough. So lactic acid is made in what type of organism, and alcohol is made in what type of organism. So I'm going to put down here under lactic acid, animals. So you've experienced this. If you're working out and you're not getting enough oxygen to your muscles, and your muscles can't continue to go this route, then they go this route, where they're still making ATP, just not nearly as much ATP. But the byproduct of that, the punishment for doing that, is you make lactic acid. The lactic acid builds up in your muscles and makes your muscles burn. Over here, plants, if they do that, they make alcohol. So in plants. And, there's a lot of people that appreciate that. Personally, not one of them. But anyway, jump over here. What if there is oxygen present? Well, then we go through two other possible steps. One of those steps, I'm going to draw it as a circle because it is a cycle, and I've been saying it all night at the wrong point, is the Krebs cycle. So one of those could be the Krebs cycle, and then when we do the Krebs cycle, we get CO2 out. So keep in mind what our equation for this is. It's C6H12O6 plus O2 gives us CO2 plus H2O. Right? Just writing that equation down for respiration quick. So we get CO2 out, And we also get 2 more ATP out of the Krebs cycle. And then once we get done with that, we go down to the electron transport chain. I could feel you guys saying that before I even wrote it. And so in the electron transport chain, we're putting oxygen, putting O2 into that. So go back up to that equation, C6H12O6 plus O2. We're putting the C6H12O6 here. We're putting in the O2 here. All right, so what do we get out of that? The other side of that equation, water comes out, and then we get a whole bunch, 34 ATP. So here's the question. How much ATP does respiration make? Does it make 36 or 34? So if you count this, if we make 2 here in glycolysis, 2 here in the Krebs cycle, and 2 here, that's 38. So just, and then some people I think don't count this. If you look it up online, it'll give you all, it will give you different answers on how much it makes. I would say if you're asked that question, you're looking for a number around 36 to 38. I hope they don't put both. I don't know what to tell you. But I hope you're looking for a number around 36 to 38. All right. That is really glycolysis. And people think this is really hard. It is not. We take our glucose, run it through glycolysis. We get a little bit of ATP out. If O2 is present, we go to the Krebs cycle and then the electron transport chain. If there's no O2, we just get those 2 ATP and we get these. So back to that question. How do prokaryotic cells get ATP? They do that. Right? They do glycolysis in the cytoplasm. They make 2 ATP and that's it. That's what they do. They can't do this because they don't have the mitochondria. How do they get photosynthesis? How does that happen? They don't have any chloroplast, but they do have chlorophyll, right? So they have that green chlorophyll substance, so they can still do that. All right, one more thing we need to draw here, we're getting close to time, is the mitochondria. So I'm going to move this a little bit just to make it easier. I'm going to draw a nice big kind of kidney bean shaped thing, and then the inside I'm going to draw that inner membrane, which is folded. And we're going to talk about why that is in a second. And so let's label some stuff here. We have our outer membrane. We have our inner membrane. What else do we have? Inside we have the matrix. One of these, like from here to here, this little where it comes in, that is called the cristae. And this whole thing, of course, is the mitochondria. Right? So we have the mitochondria. Come back over here. Where does the Krebs cycle happen? The Krebs cycle happens in the matrix. So let's add that over there. Krebs cycle happens in the matrix. Where does the electron transport chain happen? That happens on, I'm going to put the inner membrane. Could probably put the cristae there or whatever. But so the idea here is inside of this, we're doing the Krebs cycle. And then the reason that this membrane is all folded in like this is so at any spot we can have our electron transport chain happening. And what's really happening, if you take Mrs. Smith for AP Bio, you would get into this, how we're moving electrons back and forth across that, and we're using them as a kind of a pump to create energy, right? But anyway. What else did I want to add to that? Oh my gosh. Just had a total brain hiccup. All right, so you have that. We got the mitochondria, we got the cristae, got the matrix, got the outer membrane, inner membrane. What is this process called? This is respiration. Right? So this whole thing is respiration. There's our little guys, right? Our little exclamation points. So this whole thing is respiration. So, kind of sum this up. It is 8:29. If I'm shooting for a half hour, I have 1 minute. So we might go, well, it's 8:30. Let's see if I can do it in 60 seconds. Kind of sum this up. What are we talking about here? Oh, there is one other little thing I'm going to do with you guys. I want to show you. Light energy, stored energy, usable chemical energy, right? We're making our stored chemical energy here in the form of glucose. Glucose leaves that, comes over here, goes through glycolysis, Krebs cycle, electron transport chain, makes a ton of ATP that we can use then to get work done. Here's the last thing I want to show you, and I'm going to put it up on my screen. So let me change to my desktop here real quick and grab this. This was a question on your one paper pencil test. I want you to look at this equation real quick. It says glucose plus 2 ATP yields 2 pyruvic acids plus 4 ATP. And then it says 2 pyruvic acids plus oxygen gives carbon dioxide, water, and 34 ATP. So just think about that real quick. Let's jump back over to our paper. Glucose plus 2 ATP. We have to use 2 ATP for glycolysis. It yields 2 ATP, and it said pyruvic acids. So right here, this glucose, when we do glycolysis, it breaks this up into 2 smaller molecules called pyruvic acids that come down to the Krebs cycle. Once in the Krebs cycle, we add CO2. No, I'm sorry. We add oxygen, and we get out 34 ATP. So if you really think about this, glucose plus 2 ATP is the beginning of glycolysis. These are the products of glycolysis. We add oxygen to that. We get carbon dioxide, water, and 34 ATP. Check this out. If you get rid of all the nonsense, glucose plus oxygen, carbon dioxide, and water, that's respiration. So if you were given this again, and they'll do things to you like this to make you think, if you studied it long enough, you should have got, hey, if I do that, glucose plus oxygen, because glucose in this is making this product, and this product comes down here. So glucose plus oxygen, let's get rid of that, carbon dioxide and water, that is respiration. So jump down and look at the questions. 2 molecules of ATP are needed in equation A so that what? So the reaction can happen. In animals, the reaction in equation B occurs where? Well, if I know glucose plus oxygen is carbon dioxide and water is respiration, it happens in the mitochondria. Look at 34. What is the letter Y? There's this letter Y here. We haven't talked about it. Nothing's happening to it. It's not changing. It's just there. What do we know that doesn't change and is just there? An enzyme. And then what is the combined net gain of ATP molecules in reactions A and B? Net gain. Net gain. There's not 4 gained here because we use 2. So 34 plus 2 is 36. Bang. Done. I think that's it. All right. So, I think that's it. So bring this graphic organizer to me tomorrow and show it to me and get your bonus points. As always, I'm very happy you guys stopped by. I'm glad you hung out with me. It looks like we have 22 people viewing right now, which is awesome. 22 of you are going to rock this Keystone. Hopefully the rest will follow in your footsteps, but it's really great. It's really great that you can see. And I think there's a good many people starting to watch these on their own too, which I'm pretty appreciative as well. So is there any questions? I'm going to go back to the chat room because I cannot multitask. You guys think you can multitask. You're probably listening to your iPhone, Snapchatting, and doing this all at the same time. I, however, know that I cannot do that. So, the mitochondria is the powerhouse of the cell. Plants go, yes, these comments are good. Caleb, I'm sorry your cat stole your graphic organizer, but I'm glad he wants to learn about bioenergetics. That's it. That's it. No questions? You're free to go? I mean, you were free to go before. It's bonus points, I guess. If you don't have any questions, you're free to go. If anybody has questions, I'll stick around for another minute or so just to see if any questions pop up, because I know there's about a 20-second delay. So I'm talking in this screen, and you're about 20 seconds behind me. Connor Landetta, you are very welcome. This is the high point of my evening. Maybe next year I'll do this all the time and just won't come to school. Sit at home and do it. So the cat had your graphic organizer in your mouth and was running around the room. That makes it hard to write. Connor, you have no chance of me singing you a song. You'll notice that behind me are guitars, not vocal microphones. I did hint. Somebody asked at the last boot camp. I said I wouldn't know what song to play, and somebody said to sing. That's really funny. Somebody said to play Jimi Hendrix, and there's a slim possibility if you guys stick around for the last one at the end, maybe I'll play you some Jimi Hendrix. But I don't think anybody would know that anyway, so I can really play whatever I want. Mr. Suter, sing a song. I'm not going to tell you who wrote that, but she's a lovely woman. Appears that there's no questions. What guitar, Danielle? What? My guitar's at school. You know, there is no evidence that a person simply owning a guitar means that they can play it. But if you stick around to the end next time, maybe a small piece of Jimi Hendrix. Maybe. I think that would only be fair. But I gotta hold you in suspense. And so, Connor's hungry. I am hungry too. I'd like to get on with the evening. Have a wonderful night, everybody. Thank you for coming. I will see you beautiful people bright and early tomorrow morning. There's the question. Danielle, I will play you guitar for the entire home base tomorrow. It will serenade you. Have a good night, everyone. Goodbye.